something which the public, our public, all over the world, is now becoming aware of. And the reason, COVID-19. They're talking about COVID-19 mutations every day in the media, so even the general public is aware of this. They are now learning about the terminology of mRNA, the ACE2 inhibitors, and the genetic alterations of the human cell. The general public is also becoming aware of genetic testing, such as virus detection and PCR tests. As an offshoot, they're learning that certain dystrophies have a genetic basis and can be transferred from one generation to another. There were recent advances in genome sequencing and computer network technology, and this has initiated a new era in the field of human genetics. The decades of research that has shown that genetics play an important role in many eye diseases. And these genetic variants can be highly penetrant in Mendelian and non-Mendelian eye disease, meaning dominant and recessive, or they can act as risk factors in common eye diseases. The testing of genome sequencing is now available for ophthalmic dystrophies, especially keratoconus. I want to show you that I have no financial interest in any genes. But there's companies now that produce a test that can be done at home by swabbing the inside of your cheek and then sending the swab to the company for analysis. And that company now is called Avigene, and they could test for over 60 different eye genetic dystrophies. And now that genetic mutations can be identified, those who are at risk want to know if the mutant genes can be replaced with the normal gene, at least have its effect neutralized genetically. That is what this course is about. Identification of genetic mutations, what could be used to offset it, and if they occur, what could be done to treat it. A past member of this course is Dr. Aldavi, Tony Aldavi. And he recently published a study about the inheritance of certain genetic mutations in various families. Several years ago, Dr. Aldavi and I analyzed the genetics of a family with cornea plana. Now I'm going to introduce to you the faculty. We'll have Professor Namrata Sharma, who will be here later, and she'll talk about the genetics of corneal ectasias. And we have right now here Dr. Radhika Tandon, who will talk about also genetics of various corneal dystrophies. Dr. Sundararajan is now tied up with the wedding of his daughter down in Madurai, so I will be giving his talk. And finally, we have Dr. Natarajan, who did a beautiful dance last night at the ceremonies. And he's the past president of AIOS and has a tremendous interest in the genetics of the retina. I'll talk now about the Global Eye Genetics Consortium, because this consortium has been formed for the study of eye genetics. And they have a meeting now every year. It started between the United States and Japan, and now has spread worldwide. And this is from a recent meeting. Dr. Sundararajan, you can see, and Dr. Natarajan. Dr. Prakash is the coordinator from the National Eye Institute, part of the American National Institute of Health. And he works on giving grants to other countries for the study of the genetics of eye diseases. He wanted to be here today, but he has another situation in San Francisco, and he could not present virtually. In addition, there's the National Human Genome Research Institute that is part of the NIH. And they have worked on the initial sequencing of the human genome over 15 years ago. and they collaborate with the scientific and medical communities to enhance the genomic technologies. And these are some of the divisions in the National Human Genome Research Institute. It is not restricted to the eye, but they have the Genetic Disease Research Branch, the Molecular Biology Branch, and the Medical Genetics Branch that works on the disorders of metabolism and human development. Now, Tata, of course, I don't have to say it is a familiar name here in India, but he has established the Tata Hall at the University of California, San Diego. And this is a binational institute that coordinates the research between UCSD, University of California, San Diego, and India to combat the various vector-borne diseases, one of which is the COVID-19 virus. So let's start with that. This is a patient that actually is infected in the eye with the COVID-19 because there are some ACE2 receptors near the limbus of the eye. And here it is involving just the palpebral conjunctiva. Scientific American was great in the analysis and the following of the coronavirus. Here's a beautiful 3D representation. You can see the spicules of that virus in 3 dimensions. And here's the inside of it. We could see the various RNA strands and various proteins. So let's study them. We have the S proteins, the RNA is in red, the N proteins, which are in blue, and various others, the M in purple, and then there's a lipid membrane around it, and of course the E protein. So this is the mechanism. First, that virus will bind to a lung cell, and when the spoke protein latches onto an ACE2 receptor, a protease enzyme slices off the spike's head and starts the invasion on the ACE2 receptor. And here's the breakdown. The spike breaks off, fusion machinery starts, it enters the cell membrane, the human cell membrane, and it splits it, and the RNA of the virus starts pouring into the human cell. There it goes. You can see the two fused, and the RNA is pouring into it. Now, to fight it, we have the innate immune system, where an infected cell could release some interferon proteins, and it alerts some neighboring cells to create molecules that try to stop the virus particles from entering or reproducing. Interferon also calls such cells as the macrophages, you can see it in green, from entering or reproducing. And it enters the bloodstream and it can engulf the virus particles. There's the T cells that are produced that work against it. So here we have the helper T cell and the killer T cell that works against the virus. And there's the infected lung cell. The adaptive immune system, on the other hand, follows the interferon. It also alerts the B cells, and these cells produce neutralizing antibodies that might recognize parts of the spike protein and bind to it, thereby neutralizing its effect, which would be grabbing onto a lung cell. Moreover, we have antisense drugs, or at least we thought we had them when this epidemic started. We found that they really didn't work, but now new ones are coming out. And these drugs, or oligonucleotides, bind to the mRNA sequence of the virus to block the protein synthesis. And here are some of the original antisense drugs. I don't know if any of them are still being used. So let's get back to this. What we want to do, and the reason I concentrate on COVID-19, this virus could be another vehicle for delivering genetic material to change the mutants in various dystrophies. You'll see in a few minutes other vehicles that we use, but here's an additional one. Maybe that COVID-19 virus could be harnessed to deliver RNA, change the genetic structure of the diseased chromosomes, and then reproduce and create new tissue. And it might be, believe me, a type of treatment in the future. Right now we use lasers, we use surgery, especially in glaucoma, we use drugs. We're on the start of the future. This is the cusp of the future. Just look around you. It's all over you. So, J. Craig Venter, who now has a place in San Diego, adjacent to UCSD, changed the DNA software, changed the species. Here he is. He gave a keynote address at ARVO. And this is his place in San Diego. I push San Diego because that's where I live. So here's getting to basics. All of this, it's a 4-letter alphabet, not 26 letters as in English, only 4. And this is what controls all of life. 4 different letters, the purines and the pyrimidines. And if you change that and digitalize it, and that's what he wants to do, he'll convert the analog genetic code into a digital code. There it is, between the base pairs, the adenine and thymine, purine and pyrimidine, and the same with guanine and cytosine. And he worked on the mycoplasma, where he was able to change the mycoplasma capricial cell by instilling it. And this is a diagram of his where he inserted a yeast vector into a bacterial genome and changed it. And there are articles about this also, all in the New England Journal of Medicine. We have concentration of genetics. Of course, in the research journal, the IOVS, Ophthalmic Genetics that I alluded to earlier, and even our American Academy of Ophthalmology, their little magazine. And the archives, our journal, there it is. So you could, and ophthalmology, you could see how hot genetics are, ophthalmic genetics. And even for the public, here are the magazines, Newsweek, Time magazine. Want to know my future? Check my genes. Want to know what my offspring will be? Check the genes. Here's a beautiful article, Genomics on the Eye. It's not in an ophthalmology journal. It's in the New England Journal of Medicine. And scientists are regenerating retinal cells now in mice. And we're getting functional retinal cells in the eyes of adult mammals. There was genome sequencing, identification of certain mutant genes, and this way we could have specific therapy. Tissue engineering, where we'll replace, as I alluded to earlier, defective tissue with cultured stem cells. DNA gene replacement. And how is this done? I did mention gene therapy. Here's the vectors, usually viruses. We, as I said, have a new one, the COVID-19 virus. Anyway, could be genetically altered to carry normal human DNA for whatever it is, whether it's the eye or other parts of the body. Cancer is very big on this. For the trabecular meshwork, we can see the big one now is the adeno-associated virus. Others have been used, plasmids, lentiviruses, and the intravitreal AAV stereotypes usually is number 5, adeno-associated virus. And this is how it works. A single-stranded DNA genome is altered, a transgene, and a new capsid is made, and with injection, it goes from the outside of the cell into the cell. You can see the little ball on the upper left. It goes into the eye, usually with a needle, breaks into the cell, and as it passes through the cytoplasm, it goes to the nucleus, invades the nucleus, and whammo, we have a new protein made from new DNA. And ways to inject, the preferred way is now subretinal rather than vitreous, because we want to get to alter the photoreceptors and the retinal pigment epithelium. And here's some of the diseases of the outer retina. You could read them yourself. You don't need me to read it. And this is other ways of altering, especially for cancer. And the CAR T cells are extremely important. The CAR T, we make up the new T cells, and it's a combination of human and mice. That is the combination. That's what CAR T stands for. So there's the genome-wide association study that we want to change the DNA for common diseases. We can, moreover, we have not only diseases like cancer and diabetes, but the environment can also affect us and the transmission. And various genes could affect a certain disease state. Here we have LCA that could be caused by various Leber's congenital amaurosis. So we'll catalog all the proteins in the cell according to genome. It's very complex. So genetic testing, as I said, we want to predict a medical condition. Once we do that, we'll do what we can to avoid certain of the environment that could enhance the disease, and also study the genetic disease in a family. So we want to reach at a point in which knowledge of a patient's genetics could allow for development of a specific strategy for that particular patient based upon their genetic makeup. Gene therapy we're talking about for the future, and I talked about antisense drugs and vaccinations, especially for the COVID-19. So we're at the beginning. Jump ahead with us. The future is coming. Thank you. I will now introduce Dr. Tanem. Please come forth, and you're next, since you're here. Dr. Namrata Sharma is not here yet, but Radhika Tandem is. This is Dr. Radhika Tandem. Thank you.
Good morning, everyone. Thank you, Dr. Schultz, for inviting me to participate in this course and for giving such a lovely introduction. Also putting in perspective about the importance of genetics. It always seems like a very esoteric idea, and as you correctly said, the public has now realized the importance of science and the contribution of genetics. So looking at from the ophthalmology point of view, as Dr. Schultz introduced so elegantly, it's an expensive area of research. You know, it's not easy. And it's expensive in terms of resources in various formats, but it's time ophthalmologists the world over realize the importance of contributing to this area. And when we look at the advances, molecular genetics can give you a lot of information, and it can go into the nitty-gritty and take you towards the future of looking at any interventional strategies. As I mentioned, it's an expensive area, but there's a whole wealth of information available in the basic science labs, and we just need to connect the clinicians with the scientists in the lab, and then you have to work together to generate funding. Also, the public is now becoming more aware, and many of them can afford and are interested in going for commercial testing. So let's not leave that option out. Don't forget that ethics is important. So if you're going to study anything related to the genetics, even if you take a blood sample and you're going to use it for a particular study, but you're going to store those samples, and later on you may share it with another researcher, or you may do some further analysis. So it's very important when you're making your initial patient-informed consent, make it a little more broad-based if you're intending to store it and use it later on. Because purely speaking, if you have taken a sample with a particular intent and then later on you're using it for some other purpose, it would not be correct or considered ethical. Of course, conflict of interest and finances. Conflict of interest, I would mention that often we are so taken over that, we want to get ahead, we want to get the publication, but one mustn't forget that there are national interests, and contributions from Indian work must be properly acknowledged. And it's not just of doing the work, it's of building the infrastructure in our country. So whenever you do collaborate with any international team, you must engage that. You must say that, yes, at the moment we don't have this facility, but whatever the protocol is or whatever the plan is, it must integrate some element that we have to do some capacity building in our own country. We may be a little bit behind, but that doesn't matter. And then one has to look at functional analysis as well. So a little bit about cornea services. I'm going to cover a little bit about corneal dystrophies. There are unique populations in India, and I'm going to give you a few examples. For those who are not familiar with cornea as a specialty, there has been a lot of work done on the classification of corneal dystrophies with an international team. And the initial proposal, there was an idea to sort of streamline it and make it, because there were a lot of various old terms. And to credit to all the past ophthalmologists, they saw the patients, they looked at the features, they very painstakingly documented all the clinical features, and they described these disorders. And later with time, we realized that now these things need to be revised, and the IC3D classification has done that. Traditionally, it was purely an anatomical classification. You saw the patient on the slit lamp, these are the features you found, and then you classified it. However, with more information available nowadays about the actual genetics and also further the histopathology, etc., so therefore now it has been revised and you have a new classification, and this information is available freely, open source, either through the journal Cornea or just through PubMed. So the updated IC3D classification, what did it do? It firstly, it was not restricted only to the anatomical level. It excluded the Bowman's layer and Descemet's membrane from the classification. It removed the table of genes, specific genes. It reclassified CHED type 1 as PPCD. So now you only have CHED as the autosomal recessive type. It included 3 variants of epithelial recurrent erosion dystrophy, and it removed Grayson-Willebrand corneal dystrophy. You don't have to remember it, it's just to give you an example of how the classification was streamlined. Then it also categorized the information. So depending upon the level of knowledge available. So for example, category 1 disorders, the genetic locus is known, where is it, what is it, which gene is affected, what are the features of the clinical spectrum, and these are some examples. And here we have the classification. So now they are considered as epithelial and subepithelial. There are 6 of them. Then epithelial stromal. So earlier we used to have a concept that is epithelial, stromal, and endothelium. Now it's understood that there is an overlap, and largely there will be these overlaps. You're unlikely to get epithelial and endothelial or epithelial and deep stromal. So based on the clinical features, you may get epithelial stromal, then the predominantly stromal dystrophies and the endothelial dystrophies. So looking at the concept, we had also had a low vision services is something which has been developed, and apart from retinal disorders, even corneal disorders are responsible for low vision. And it was felt that whatever we do, ultimately it has to be for the benefit of the patient. And certain disorders, of course, they can be easily corrected by surgery, but still it's always good to prevent the disease, because surgery is not a very uniform or an excellent solution, particularly for diseases like CHED. So along with the idea of looking at strategies to restore vision, it was also to have a forward-thinking approach to look for interventions for the various genetic disorders. So we had formed an action plan, think tank, and a brainstorming session. And basically we identified certain disorders which are important, worth working on in India, where we felt that either they are given orphan status, they're not that relevant internationally, or we may have unique populations in India, or we would really like to know what is happening with the population in our country, because there may be a little bit of variation in the genetic workup. So we had the Asian Eye Genetics Consortium, which was set up, and the idea was to look at the study of genetics of ocular disorders in India. There is a whole group of people interested. And then, of course, there were diseases in the cornea, glaucoma, optic nerve retina, and retinoblastoma. So I'll give you a little example of work that we have done over the years looking at corneal dystrophies. Dr. Arundhati Sharma is the head of our genetics lab. So as you know, the corneal dystrophies are inherited disorders. And looking at our spectrum of disorders in the North Indian population, we found that in our population we had autosomal dominant, that is the granular and lattice were the predominant. Then we had the macular dystrophies, we had CHED and Fuchs. This is the distribution in this particular cohort of patients. The other interesting thing is the age of onset of granular dystrophy out of 92. As you can see, it traditionally is described as not being that early onset, but we didn't have people in the first decade with granular corneal dystrophy. As far as lattice is concerned, we had no patient with onset in the first decade, and macular dystrophy is traditionally known to have an earlier onset. So this is showing largely all the patients presented early rather than late. And here I would say the difference is because in textbooks the description is based on the Western populations. And what happens is because of rural or very extensively peripheral location of patients, or even the concept of the patients, they may not seek intervention immediately or early on. So this is our example of the genes that we found. We did find certain new genes which were first reported from India and certain novel mutations. And sequences, this is some example of our sequencing data and some examples of the new cases. Novel mutation, granular corneal dystrophy, and here again there's a variation. This is granular corneal dystrophy as well as Avellino corneal dystrophy. And these are certain examples of mutation. This was a variant of lattice corneal dystrophy. And this is another example of a novel mutation which has been described from our patients. The clinical photographs were suggestive of map dot fingerprint, but when you looked at the siblings, they had the classical evidence of granular dystrophy, and what turned out was a novel mutation. Similarly, for macular corneal dystrophy, this is an autosomal recessive stromal dystrophy. Here again, we were able to describe some novel mutations, and the molecular genetic analysis concluded. So we found, as our conclusion, that there were 32 patients of macular corneal dystrophy. Some of them we were not able to identify any specific mutation. Coming to the endothelial dystrophy, we had Fuchs and congenital hereditary endothelial dystrophy. 39% of them were familial, and the male-female ratio, though corneal dystrophies are said to be equal in males and females, but we have to see it will depend upon the patients who are seeking help and actually coming to the hospital. So a hospital-based distribution may not be absolutely accurate because there may be a gender bias in seeking care or reaching to a tertiary care hospital. Fuchs, and then this is posterior polymorphous. And then we had some variations, very interesting. A patient of CHED who actually presented as late as 30 years of age. The patient was managing all her activities with that poor vision. So therefore had further changes which are not normally associated with CHED because the patient had been with it for so many more years. Iron lines, spheroidal degeneration, and rare occurrence of bully, which are not normally seen. And this is some examples of Fuchs dystrophy. And ASOCT can show very characteristic features in Fuchs. You can have thickening of the desmoids. You can have subepithelial cysts in CHED with spheroidal degeneration. And you can see gutted changes in Fuchs dystrophy on ASOCT. And these are the confocal microscopy examinations. And then histopathology also, there was appearance of the normal cornea, CHED and Fuchs. Again, here they can be very distinct, which is interesting because all of them are actually manifestations of an endothelial dysfunction. The difference being the endothelium is often absent in cases of CHED, whereas in Fuchs it is present, but it is not functioning normally. And as far as the molecular genetic analysis of CHED, we had 35 subjects from 28 families. All the 19 coding exons of SLC411 flanking splice junction, the putative promoter regions were amplified. Bidirectional sequencing was done. Clinical and histopathological findings were compared with the molecular genetic findings for genotypic-phenotypic correlations. Compared to the other disorders, CHED had a relatively lower proportion of cases. There was a higher degree of consanguinity which was seen among the patients of CHED, but the pattern of consanguinity in North India is more among cousins across the same generation rather than vertical. And here again we were able to report a novel mutation. But still, no mutation could be identified in 21 patients in the identified coding region. So definitely there is an existence of genetic heterogeneity, a kind of an overlapping of cases, which could be worth studying for the future. And the molecular genetic analysis for Fuchs again, we look for the described COL8A2 gene. A total of 30 subjects were recruited, and I'd like to acknowledge the Department of Biotechnology, which funded this work, Department of Molecular Genetics, which is run by Professor Arundhati Sharma, and Dr. Preeti Paliwal, who is a PhD student. Cornea Services, we have several corneal surgeons who contributed patients, ocular pathology by our ocular pathologists, and the Department of Anatomy, which helped us with the electron microscopy work. And these are our publications. Little bit more about CHED. I mentioned CHED, and this is where it is important that we continue the research, because if this could be prevented, or if we could have a prenatal or an antenatal diagnosis and one could take an intervention in utero, that would really be helpful. So here there's corneal clouding present at birth, there's blurred vision, there's nystagmus, there's a diffuse ground glass haze and an increased corneal thickness. And you can have secondary changes, like I explained earlier, if the patient lives with the disease for several years. You can have band-shaped keratopathy, spheroidal degeneration, and so on. Now, what do we know about the genetics? It's present on chromosome 20p13. The mutations are in the SLC411 gene, which encodes a membrane-bound sodium borate cotransporter. Now, looking at this was the example of the patients I showed, histopathology, and there is a thickened desmoid and an absent endothelium, as opposed to Fuchs, where the endothelium is present but non-functional. And you can see corneal edema and epithelial bullae on histopathology, which are not that much seen clinically. Now, giving you an example of overlap or confusion in diagnosing the child at birth. So there was a patient who presented. There was a demography. There was a 6-year-old female who'd come from the state of Bihar. The father was the informant, and the chief complaint was whitish opacity noted since birth. But please note, the child has had this opacity, but the parents actually were able to come for medical attention when the child was 6 years old. A large size of eyeballs was noted. Involuntary eye movements were noted. There was no history of watering, redness, or photophobia, and no history of trauma. I'll quickly go over. So this was the family history, the examination under anesthesia. This was the findings that we found. And in such a patient, we always have a differential diagnosis of glaucoma because of the enlarged eyeball size. But this could be sometimes secondary glaucoma because and leading to a raised intraocular pressure. There were no other contributory features. Glaucoma surgery and keratoplasty was planned. This was the ASOCT images. These were the images in ultrasound biomicroscopy and the investigations. Go slowly? Go slowly. Okay. So this was the differential diagnosis. Whether it's primary congenital glaucoma with a leukomatous corneal opacity or an anterior segment dysgenesis with secondary glaucoma. And we're very fortunate at the RP Center because we have a multi-speciality teaching tertiary care referral hospital with experts in every field. And we often take help of our glaucoma experts for care of such patients. So this was the histopathology. So corneal opacities in children, the differentiation becomes difficult, whether it's CHED or non-CHED, acquired, traumatic, or congenital glaucoma. And plus we have the spectrum of disorders of Peters anomaly and anterior segment dysgenesis. So look at this publication by Dr. Ramamurthy, coexistent congenital hereditary endothelial dystrophy and congenital glaucoma. Retrospective evaluation of 10 infants with diffuse corneal edema and bilateral raised IOP, and histopathology showed diffuse stromal edema with loss of the endothelial cell layer and thickening of posterior non-banded portion of Descemet's membrane. Looking at Peters anomaly for our patient, points for, points against, the presence of, so basically that's sometimes difficult to differentiate mesodermal dysgenesis, and looking at the genetics of congenital corneal opacification, the impact of diagnosis and treatment, another very useful paper by Ken Nischer. So now looking at our patients, we thought that we would also screen them for these disorders, exploring the application of artificial intelligence in combination, with the idea being that is it possible to have a kind of an algorithm to help us differentiate the patients right in the beginning. So just a snapshot of the preliminary results. 42 patients, 82 eyes were included for analysis, and we had a significant difference in the ability of the artificial intelligence to actually differentiate. The accuracy was excellent. And looking at the genetic analysis of the patients, we were again able to find a novel mutation in the CYP1B1 gene. And in all three groups, we have found good results. So I just want to leave you with this to say that it's extremely important that we do these studies in our population so that whatever interventions are possible in future, our patients or local patients can benefit. Plus, we can also contribute a lot to the international forum because of the expertise available in our country, and capacity building and motivation of clinicians to join hands with basic scientists is really important. Thank you so much.
Are there any questions? This was a fantastic analysis of the various corneal dystrophies. And what we have now, as you saw, is we don't have to have path specimens to make the diagnosis. We have anterior segment OCT. We could do it on a live patient.
Yes, that's true.
So please ask your questions about anything on the corneal dystrophies, the inheritance and the treatment. Go ahead, please.
Thank you very much for the presentation. My question is, in situations where you are unable to do the genetics, and you know that happens a lot in some environment like where I come from, and apart from, you know, some of them may have raised intraocular pressure without the corneal enlargement, and the increased IOP is largely due to the very thick corneas that they have. How do you then differentiate between those that are due to glaucoma, PCG, or those that are due to CHED? I mean, you can have a child who has bilateral congenital glaucoma, and you can have a child with CHED. So how do you differentiate between these two groups of patients? You know, because I think it's important. Or do you just do a blanket treatment, start them both on anti-glaucoma therapies for people like us who don't have access to genetics, you know, at least before you get that?
Yeah, very nice question and very pertinent. So we don't rely on genetics to make the diagnosis, but we do use the information that we have derived from genetics. So one could do a careful family history, for example, and also look at other family members. The other point being about differentiating. So as you correctly said, ultimately what matters is the vision of the patient, and the control of intraocular pressure is extremely important. Estimating the intraocular pressure is a challenge because, as you said, the cornea may be thick and it may appear high, it may be low. So we don't rely on only one parameter. One may look at the digital intraocular pressure. One would also do an ultrasound for optic nerve head cupping, do the globe size, and do it along with a glaucoma expert. Now the question is whether they have, because in pediatric age group, medical treatment for glaucoma is not usually recommended unless you're in the phase of a secondary glaucoma and you just want to control the pressure and you know, combine that with the keratoplasty. So practically, you'll have to break it down into what is essential at that particular point of time and then go accordingly.
Do we have other questions? If not, I will now introduce Dr. Natarajan, who will be talking on the genetics of retinal dystrophies and diabetic retinopathy. Perfect timing.
Thank you, Gerald. And Jerry had his birthday yesterday, and we did a cake cutting. And he also makes good cakes, right, Jerry?
Excuse me?
You also make good cake, no?
You got it right. And he's a fantastic dancer. These are skills. I've known him for decades. He keeps coming up with new things. Fantastic dancer and dresser.
Thank you, Jerry. And I think if you, he has brought on, oh, he's here. So he's got a resident from, you're from USD, right? Welcome. So my talk is on genetics of retinal dystrophies and diabetic retinopathy, and I have no financial interest or to disclose. So genomics and the eye had a pivotal role in the evolution of human genomics. At least 90% of the genes in the human genome are expressed in one or more of the eye's many tissues and cell types at some point during a person's life. And this has also been, this is published in this review article, but it's also been mentioned in the Sorsby's book, which he calls Ophthalmology is a Playground of Genetics, which I read in 80s when I was a postgraduate. And I think still, I remember that time, my father was a professor there, and he said 50% of the Parsons, they really you can't do anything. And I'm glad the rest of the 50% only we are working on now. And whatever we thought could not be done, I think that one of the examples is the macular dystrophy, which is characterized by a progressive central visual loss and degeneration of the macula and underlying retinal pigment epithelium of the eye. So it's and then the other is autosomal recessive, Stargardt's disease, and fundus flavimaculatus. So Stargardt's dystrophy is the most common hereditary macular dystrophy and accounts for 7% of all retinal dystrophies. Unfortunately, we have one of the largest group of patients in the world, and we actually applied for a research grant from Medical Research Council UK for almost 21 crores along with the Queen's University for a new drug treatment for Stargardt's, but anyhow we didn't get it. But still a lot of work is going on. Typically patients present with decreased central vision, usually in the first or second decade of life, characterized by bilateral atrophic changes in the macula, degeneration of the underlying retinal pigment epithelium, and the presence of prominent yellow-white flecks in posterior pole. The locus of the Stargardt's was mapped to gene ABCA4 on chromosome 1p, and ABCA4 is involved in ATP-dependent transport of retinoids from photoreceptor to retinal pigment epithelium. Failure of this transport results in deposition of major lipofuscin fluorophore in the RPE, secondary photoreceptor degeneration. And the two chromosomal loci have been identified, and the other is the best disease, the vitiliform macular dystrophy. Patients are found to have bilateral macular lesions at a very young age. Typical fundus finding including a yellow egg yolk-like appearance of the macula. And incidentally, today there is a program by the National Eye Institute organized by John Prakash, the patron of the Global Eye Genetic Consortium, along with Takeshi Iwata, the president of the Global Eye Genetic Consortium, and myself. We have a program in the US, which is, I think, 6:30 p.m. India time and 10 a.m. Eastern Standard Time. So it's on genetics again. Typical fundus findings including a yellow egg yolk-like appearance of the macula. So VMD2 gene on chromosome 11q is responsible for maintaining chloride conductance across the RPE. Its defect leads to accumulation of fluid and or debris leading to detachment and secondary photoreceptor degeneration. See, the other disorders are adult vitiliform macular dystrophy, pattern dystrophy, Doyne honeycomb retinal dystrophy, malacia leventinus, autosomal dominant drusen, autosomal and macular degeneration, North Carolina macular dystrophy, progressive bifocal chorioretinal atrophy, Sorsby fundus dystrophy, central areolar choroidal dystrophy, and dominant cystoid macular dystrophy. And all these I think are required because we are in the time of doing the phenotype and the genotype. And I think the problem is even phenotype, many people, patients at least in India, don't want to get the ERG or any of the, even the genetic testing, because if there's no treatment. But I think we have to create awareness. and this is a X-linked juvenile retinoschisis. The XLRS is a vitreoretinal degeneration which presents either in an infant with nystagmus or more commonly in childhood with mild loss of central vision. The characteristic fundus abnormality is a cystic spoke wheel-like maculopathy in virtually all affected males. Prognosis is good in most affected males as long as retinal detachment or vitreous hemorrhage does not occur. It has been linked to XLRS1, also recently referred to as RS1 XP22. It is implicated in cell-to-cell adhesion and cell-to-matrix interaction, which correlate well with the observed splitting of the retina in the XLRS. And there's a clinical trial going on in the NEI for the even retinoschisis by the former director who's now moved to UC Davis. This mitochondrial inheritance, maternally inherited diabetes and deafness. This mitochondrial DNA mutation may be associated with a severe encephalopathy with death at a young age. Macular pattern dystrophy has been found in association with the MIDD. As the prevalence of MPD in MIDD is high, the association of MPD with diabetes should raise the possibility of screening for a mutation of mitochondrial inherited problem. So this is a picture from the genetics of the inherited macular dystrophies. And the diabetic retinopathy, we have a major burden, but still the genetics is complex, and the potential candidate genes include the various list which is mentioned, and a lot of work is being done. Okay. And this is the work done by Shankar Netla group by Rajiv Raman and Professor Kumar Manikvel and Uttra. And here in the aldose reductase, chromosome 7q35, it has been strongly implicated in the pathogenesis of microaneurysms and degeneration of retinal pericytes. Z2 is a high-risk allele for diabetic retinopathy, especially proliferative diabetic retinopathy and maculopathy in Indian patients. Again, the work done by Professor Kumar Manikvel. The vascular endothelial growth factor, chromosome 6p12, high vitreous levels have been repeatedly detected in eyes of patients of proliferative diabetic retinopathy and diabetic macular edema. VEGF inhibition has been shown to ameliorate retinal changes, including retinal vascularization and breakdown of the blood-retinal barrier. The angiotensin converting enzyme, a recent independent meta-analysis suggested that ACEID, that the insertion deletion, may be associated with proliferative diabetic retinopathy under a dominant genetic model, assuming either fixed or random effects. Autoantibodies to glutamic acid decarboxylase show an inverse relationship with the retinopathy. The protective mutations in the Asian Indian population, aldose reductase ALR2 alleles 200 and 220 BP protect from developing retinopathy. That's what is a big problem, because they are the people who, if you don't identify, they say we don't control diabetes, but still we don't have retinopathy. So I think people don't understand the genetics in diabetic retinopathy. The SER82 allele in the receptor for AGE, the RAD gene, is a low-risk allele for developing diabetic retinopathy in Asian Indian patients who have type 2 diabetes. The other retinal degeneration, the retinitis pigmentosa, it can be inherited in an autosomal dominant, autosomal recessive, or X-linked manner. So the patients with RP are many times in depression because they don't understand. Sometimes they are only one person, the whole family having it, and the other will be in an autosomal dominant or recessive. So you have the Leber congenital amaurosis, autosomal dominant, and autosomal recessive, and you have autosomal. So this is the number of identified genes, and I think there are about 200 genes which are associated with the RP. And DNA testing is available on a clinical basis for RP. And the main thing is the discovery that ABCA4 is involved in transporting vitamin A derivatives out of outer segment disc led to the discovery that vitamin inhibitors such as fenretinide inhibit the accumulation of lipofuscin in animal models of Stargardt's disease. The fenretinide is not available easily, and that's a trial which we wanted to do, but it was a huge amount, so we couldn't do that. And the identification of the role of VEGF in CNVM led to the development of therapeutic antibodies to combat the major blinding complication of AMD. And growth factors and neuroprotective agents have also been used to reduce the rate of an apoptic response to inherited cellular abnormalities. In recent years, gene replacement therapy therapeutic stem cells and retinal processes have also moved to the threshold of clinical use for the treatment of genetic eye disease. And the mechanism of gene transfer, either through the adenovirus vectors, adeno-associated virus vectors, herpes simplex virus vectors, RNA virus vectors, physicochemical gene transfer, the lipoaffection, and others. And these are the lipid-mediated gene transfer. And therapeutic approaches you have the ribosome therapy, antisense therapy, suicide gene therapy, growth factor therapy, gene replacement therapy. So I wanted to say that in RP, if you have a RP65 mutation, we have the Luxturna. I don't have any financial interest, but unfortunately, it costs 6 crores for an eye. But the clinical trials are going on, and it's an FDA-approved gene therapy. And there are several things are in the pipeline, and I think in the RO, we had several newer genetic, and unfortunately, most of it is going to come to vitreoretinal surgeons because we have to inject the gene vector into the subretinal space near the macula. And there is a new development where robot has been developed in Ghent, Belgium, and in Switzerland by Dr. Mark D. Smet, where we can inject, because the problem is to inject in the subretinal space without getting the genes out of the subretinal space. So there's it's being done in Belgium, and hope that it'll become a standard. I know it's going to be expensive, but still it'll be with the vitreoretinal surgeon's armamentarium. And it has helped refine diagnosis, disease classification, prognosis, and improved genetic counseling, which is still growing in India. Thank you.
Thank you, Dr. Natarajan. If you have any questions, please ask them now for Dr. Natarajan. Please, go ahead.
Thank you. Just curious, when you were talking about the mitochondrial-induced diabetes, for Indians, you said, surprisingly, they don't have retinopathies.
No. Only one group.
Only one
if
group.
If they have that gene, it's a protective. We still don't know. A lot of work have been, the numbers are small.
Okay.
Because genetic testing is expensive, and it's like chasing the genes in an ocean, and we don't know which one is protective still.
Yeah,
Even now, we are writing an RO1 grant for the US with the.
That's not even the question. I just want to link it to glaucoma, because I'm in glaucoma. And as part of the glaucoma pathogenesis, you talk about the nitric oxide synthase anomaly, the other things, the TNF, having the genes mutation. And you also know that there's a controversy as to whether diabetes leads to a worsening of glaucoma or even development of glaucoma, the association, there's a controversy. So my question is
we'll be talking about glaucoma later, the presentation on glaucoma.
Okay, so I can leave the question until then?
Yeah.
Okay.
Because we have to move on.
So, so there are two parts. One is the inherited, I'm just answering. Inherited retinal degeneration, we can find the specific gene. And diseases like glaucoma and diabetes, it's like a massive, so a lot of research is going on. There's a huge grant, 11 million grant to Shai's Eye Institute in the UPenn, where they're working on the glaucoma, and particularly African Americans.
All right. I now want to introduce Dr. Namrata Sharma, who will be talking to us on corneal ectasia. Dr. Sharma, as we all know, is the secretary, wonderful job, of the AIOS. Unbelievable amount of work you've been putting in, totally. Dr. Sharma.
Thank you, Jerry. Thank you for your visit here to India. It's always a pleasure to have you here. And thank you for making me a part of this very important course in genetics. I would be talking to you about genetics of corneal ectasias. And the causes of corneal ectasias could be primary or could be iatrogenic. And genetics does have a role even in iatrogenic cases, because if something is linked genetically, then the propensity for ectasias after various surgery increases in these cases. Keratoconus is the most common form of ectasia that we see, and in all the literature that is available, it is generally tends to be bilateral, although it is asymmetrical. But it can be unilateral, and surprisingly, one study showed that 41% keratoconus patients were unilateral during the diagnosis. We currently have this ABCD classification of keratoconus which we follow. An important thing to know from this diagram is that there are various types of collagens which are present in various layers of the cornea. So type 1 and 5 are present in stroma, 1, 3, 4 and 12 are present in the Bowman's, and 4 and 7 are present in the basement membrane. And these essentially become important when we are looking at genetics of keratoconus. There is reduction of epithelial and stromal keratocytes. If you see, if you look at the molecular level, there's reduction of epithelial and stromal collagen content, degradation of corneal membranes and extracellular matrix, abnormal mitochondrial functions, and even deranged lipid metabolism. So there's upregulation of the degradative enzymes, which is expected, and there's downregulation of the proteinase inhibitors. 4 times increase in the receptors of interleukin-1 occurs, which causes apoptosis of the stromal keratocytes, and there's also microtrauma, which releases the interleukin-1 from the epithelium. So if you look at the etiology of the keratoconus, this summarizes it very well. There is a molecular aspect to it, and there is also a mechanical aspect to it. So molecular hypothesis, according to the molecular hypothesis, there is unknown collagen dystrophy that is there. But when factors like atopy, eye rubbing, etc., and inflammation are coupled with it, then it leads to having keratoconus. And there is purely also a mechanical theory to it, where eye rubbing causes changes at the molecular level due to inflammation, and that causes keratoconus. And this essentially summarizes what I was talking about. So if you look at the genes, there are various genes which are cornea-associated genes, like corneal biomechanics, central corneal thickness, keratoconus genes, corneal dystrophy genes, and the corneal curvature genes, and various ocular syndromes also. And I think some of them become important, like this COL-BA2 gene, which becomes important because these are overlapping genes. So just to show, like various ocular syndromes again are overlapping genes with the keratoconus, and that is why some of the syndromic syndromes would have keratoconus. And keratoconus specific genes with no other ocular disease are as shown, and I think LOX is the one which is most important, and there are others which have also been isolated. Now there are various techniques for mapping these genes, which include the standard linkage analysis, the GWAS, the NGS, or the next generation sequencing, copy number variation, and mutational analysis. And this is our study, which was done at our center, mitochondrial complex 1 gene analysis in cases of keratoconus. And it did show that there are 18 nucleotide variations which are studied, and 2 novel frame shift mutations were detected in this cohort of 20 keratoconus cases when it was compared with 20 healthy eyes. And this is the picture from the same, where there was deletion of nucleotide G codon change from GGC to GCT, and deletion of nucleotide CA codon change from ATC to ATT. This was another study where we looked at VSX1 gene analysis in cases of keratoconus, and no pathogenic VSX1 mutation was identified in this cohort. But there were 2 alterations of VSX1 gene which are noted, and both variations were non-pathogenic. And other genetic loci such as 13q32 may have been involved in cases of pathogenesis of keratoconus, was something that again we concluded from this study. Then candidate genes which are associated with keratoconus are as shown: the VSX1, the MIR184, the DOC9, SOD1, interleukin-1, COL4A3, 4A4, and 5A1, which are primarily the collagen genes. And this helps you to know the disease mechanism. Once you study these genes, then one can identify the biomarkers and also correlate it with a family history. So the genes which are associated with keratoconus, these are the functions of those candidate genes, and genetic conditions are also associated with keratoconus, and this is a whole list of the conditions which are associated with it. If you look at the inheritance pattern, it could be sporadic or it could be familial keratoconus. 90% of the pedigrees with familial keratoconus display autosomal dominant inheritance with reduced penetrance. And then there have been studies also on the monozygotic twin studies where there's higher rate of concordance, greater evidence for primary genetic causation rather than environmental etiology. And the concordance does support evidence of heredity as a genetic factor in keratoconus. And in cases of discordant for keratoconus reading, it was the other things, that is the environmental factors which are responsible for eye rubbing for etiology of keratoconus. Then again, this is a study from Narayan Netralaya where they looked at the inflammatory markers and they found high levels of inflammatory factors such as TNF-alpha, interleukin-C and MMP9 and altered corneal epithelial and stromal expression of the specific genes at corneal cone apex were there which drove actually the focal structural weakness in keratoconus. Now, this is the list of the identified genomic loci in keratoconus through various linkage studies, and loci 5q is being replicated in different studies, so I think this remains constant, although all the other factors are a little variable. And keratoconus susceptible genes were reported in different studies based on the different techniques. So that could be one confounding factor, and this is again a list of the various genes which have been identified with keratoconus. So overview of all the investigated genes in keratoconus is highlighted in this diagram, which was published in a review article, and I think the LOX is the one which is the most important. Now, genetic screening does help in these cases. This was a study of 79 Chinese and 9 Greek families of keratoconus, where 91 patients were detected or diagnosed to have keratoconus, and there were 5 variants in VSX1 and TGF-beta1 genes which were identified in these families. So there is no causative gene known for keratoconus that causes the disease directly, and susceptibility genes have to be identified with about 200 polymorphisms in more than 50 genes. Available genetic information as such now is insufficient for keratoconus, and there are no specific pathways which have actually been confirmed. A word about keratoglobus: nothing much have been done by in other ectasias. Recent study reported association of variants of this with TMEM45A with keratoglobus, and with pellucid marginal degeneration also, which is a rare disorder. Hardly any studies are available, and only COL5A1 mRNA expression was significantly lower in keratoglobus and PMD keratocytes, and again LOX mRNA expression was higher in patients with keratoglobus keratocytes. So like I said, we need to still study a lot. There is a lot of variation, lot of variability, and lot of overlaps and interlaps which need to be sorted out as far as the genetics of keratoconus is concerned. Thank you very much for your kind attention.
Well, thank you, Dr. Sharma. That was a fantastic in-depth analysis of all types of corneal ectasias and the genetic basis. Do we have any questions for Dr. Sharma? Please, this is your chance from the authority. Thank you, Dr. Sharma.
Thank you so much, Sherry. Thank you.
All right, the next talk will be on genetic alterations in glaucoma, being given by Dr. Schultz in place of Dr. Sundararajan, who was tied up preparing his daughter for a wedding in Madurai. As an aside, I have prepared a paper for those on the definitions of the genetic terms. So if any of you want a copy of this, and it's an in-depth on the definitions of all the terms that we've been using. Unfortunately, a lot of the terms, we just use the initials without telling you what it is. For instance, does anyone know what the CAR-T is? What is CAR-T? Okay, so much for that. That's the chimeric antigen receptor that is a combination of human and animals put together for preparing T cells anti-inflammatory. What is CRISPR? C-R-I-S-P-R. Okay, what is CRISPR? Okay, It is clustered, regularly interspersed short palindromic repeats, a family of DNA sequences that when we cut it, we can alter the genetics of chromosome. And with that, I'll now give Dr. Sundaresan's talk. What I will do is ask those that would like a copy of this. I have a pen and paper here. Put down your email address, and when I get back to the United States, I will then send you a copy of this. We'll do it at the end of this session. We're on. Dr. Sundarasan. And we'll start from the first one. And we'll talk about the molecular genetics of glaucoma. And this is an amazing field because everyone talks about how could we alter the genes? What gene is causing glaucoma? Because glaucoma is so prevalent. Can we attack, can we change one or more of the genes? Well, let's find out. And this should answer some of your questions too. All right, so primary open-angle glaucoma is a very complex neurodegenerative disorder. Has anyone considered that glaucoma could be a neural function? Thinking of pressure, could it be neuro? Well, it is because it affects the optic nerve. It definitely is neuro. And we've got 542 genes with confirmed associations with glaucoma and its related phenotypes. Normal tension glaucoma, ocular hypertension, open-angle glaucoma, primary congenital glaucoma, so many of them. So let's take a look at this, the genes and the phenotypes. Well, we know that a gene is a region of DNA coding for a protein, and with that we get a disease phenotype, and we'll go into some of these, because that's a trait produced by the interaction of genes and the environment. And the endophenotypes, so-called intermediate phenotypes, it doesn't really come out that much. Under B, we have genes causing the primary open-angle glaucoma. Now we're getting specific with the eye. So what do the genes do? Well, it's responsible for intraocular pressure, the disc area, and the cupping. But the cupping is not only the horizontal, but it's the depth of cupping as it hits the lamina cribrosa. And we see that especially now that we have OCT again, and we can see the OCT of the optic nerve, how deep it is. Anyone here know about Starbucks? Is Starbucks prevalent in India? Because for those that know, it's like a vente. Some of those discs, like a vente, that is a deep, broad cup of Starbucks coffee. So let's take a look at the genes causing normal tension glaucoma. Okay, there are 5 of them, and high tension glaucoma, that really brings it to our attention when the pressures go into the 20s. And finally, glaucoma in general. And here's the beautiful overlap. We saw one of these from Dr. Sharma, where we get the overlappings. These are the genes implicated in primary open-angle glaucoma and the optic disc. Cup-to-disc ratio, we have about 5 genes, just for the cup-to-disc ratio. And for the cup area, about 11 genes. But we have 8 genes that overlap. And now if you take the disc area with 9 genes, we still have a couple of loci that overlap, and we've got 3 of them that overlap everything. So you can see that it's a very complex issue for the broadness of the disc, the cup area, and the cup depth. Now let's go into the genes that are implicated in primary open-angle glaucoma and the pressure. For intraocular pressure, there are about 7 of them, and for central corneal thickness, And believe me, corneal thickness has a lot because if it's a weak cornea, you're not going to get that pressure in the eye. If it's a stiff cornea, a little bit could build it up. And also, when you're taking the pressure, you have to also really take into account the thickness of the cornea in high myopes or high hyperopes, because that could throw you off on your readings, whether it's applanation or the Schultz. Does anyone know what the Schultz tonometer is? Great. Great. Because that is more accurate than those mechanical digital devices. I know because I run various methods of taking pressures in my office, with the gold standard being the Goldman applanation. And I'll tell you, these machine-oriented digital ones, they're terrible. Okay. And we can see that we have, for intraocular pressure, 7 genes, and for central corneal thickness, 28, with a couple of them, one of them that overlaps. And here's another diagram of overlapping. Normal tension glaucoma, we can see that in yellow. Yeah, there are about 52 genes, but look at the overlaps with ocular hypertension and primary open-angle glaucoma, and also the juvenile open-angle glaucoma, JOAG. I hate these little initials because unless we define them, we don't know what we're talking about. So genes are classified according to their association with the ocular tissues and the phenotypes that I alluded to, and their functional annotation and the damage that's done. So there's special primary pathways. We have to analyze the lamina cribrosa. Of course, that's where the nerves get crushed. The optic nerve tissue gene analysis. The optic nerve hypertrophy, optic nerve head astrocyte gene analysis. Someone asked the question about the retina and gene analysis and the trabecular meshwork. So you could read down here, open angle, normal tension, and the juvenile. And these are the specific pathways. Look at the number of genes for the lamina cribrosa that could cause problems. 69 of them, and the optic nerve tissue gene, 62 of them. Ocular nerve head astrocyte genes, and the retina causing it, all the way down to the trabecular meshwork. And of course, ocular hypertension, which can be a potential if it goes high enough for causing glaucoma. And our main concern, of course, is juvenile open-angle glaucoma. So there's no single molecular pathway. Ooh, look at that tricky finger. There's no single molecular pathway that can encompass all of primary open-angle glaucoma. And there's inflammation, that's senescence, aging, inflammation, especially if it affects the trabecular meshwork. So transforming growth factor, again, we're using initials. Transforming growth factor B is a gene, and it's a signaling pathway that could be implicated in the analysis of glaucoma. And a certain one of these, transforming growth factor B or beta, and particular research attention, because it could be elevated in the aqueous humor and of primary, but not in the other forms of glaucoma. That's one of the genes that could produce a substance in the aqueous that could affect the trabecular meshwork. So here, this is a lot of reading. It's a terrible slide because there's so much in it, but let's go through it. Transforming growth factor is a signal for the tissue, and regulates a lot of the cellular processes, cell growth, migration, differentiation, et cetera, et cetera. And with that is another gene, the SMAD. How it got its name, I don't know. But that's the SMAD, that's another gene. So the transforming growth factor, this is in red, and the SMAD pathway is of interest because there are some functional studies that shows that the signaling of it can develop pathologic changes with primary open-angle glaucoma. And now we have another gene, CDKN2B, also has a regulatory role, and the SSAS1 could regulate that, and that could be an enzyme. So we could see that the elevation of it could trigger glaucoma in older people. Let's not call it senescents, just call them senior citizens. Oh, forget, nothing personal. So the contributions to the genes, we have the MYOC, MYOC, that's a gene, a very primary gene, and how it's expressed. And there are a whole bunch of projects, DBT, I don't know what it stands for. I asked Dr. Sundaresan what are all these initials for the projects mean. I didn't get an answer. But we could see that that there's genes in particular projects that have been studied. Then we have the six gene, SIX6. That's another gene responsible. These are the projects and groups that are studying various genes and their effect. And myelin is the myco gene causing myelin, and that gets into the aqueous too. And look at all the genes that produce this. It's huge, huge, because that's another factor in producing elevated pressure and the damage. This is a paper on the common invariant of transforming growth factor associated with primary open-angle glaucoma. I left this in because at least it talks about primary open-angle glaucoma. Now this is going to be a very interesting set of slides because various regions of India have different subtypes, different genes and interactions. Just watch this. This is a breakdown of the states of India. Here, the MYOC gene affecting certain regions, causing primary open-angle glaucoma. And here's the one with the CYP1B1 gene and the effect and where it could be found. And finally, for primary open-angle, for chronic pediatric glaucoma, the gene that's affected, and it's just a few regions with pediatric. And we can see the genes and its loci all over India. With the different populations, as we know, different languages and different ethnics, they will have different genes that could cause glaucoma. Now here's a deletion in the regulatory region of SIX6. I don't know how it got its name. I think the person that named it stuttered. But it's a risk factor for open-angle glaucoma, and Dr. Sundaresan was one of the investigators. And here's the location of single nuclears that were identified and how it could be altered. This could be altered, and some types of glaucoma worked on. So here's the effect of some deletions. All it takes is a deletion, and you can change the enhancer of a gene causing glaucoma. Remember I asked about CRISPR? Well, CRISPR Cas9 is the protein that works with the CRISPR, which is an amalgamation of the clustered regulatory interspace short palindromic repeats, and how working on this could also alter with the myelin glaucoma. So here's some current testing recommendations for genetic analysis. Juvenile open-angle glaucoma, we're producing a myelin that could affect pressure, and we can see the terrible cupping there on the juvenile, and primary open-angle glaucoma, and we could see the effect on the visual field. So in conclusion, primary open-angle glaucoma, it's very complex. It is a neurological disease. Think of it that way. And many genes are implicated in it. Studies have shown that inflammation and the older age group can play pivotal roles in this. And apart from the administration of pressure-lowering drugs, we hope that we could develop novel and effective therapeutic interventions genetically to tackle the neuroinflammation the tissue remodeling, and eventually hold off blindness. Thank you very much. I'll try to answer the questions from my basis of the genetics of glaucoma. Yes.
Thank you very much. Now, my question is the basis of the neurodegeneration in glaucoma with regards to the genetics. Now, there is the link with Alzheimer's and Parkinson's disease with the tau proteins and those other things. I didn't see that mentioned here. I think it's the APPB lipoproteins, the...
For juvenile glaucoma?
Yes.
Well, at least where we are, we don't see that much juvenile glaucoma.
Oh, we see a lot.
Maybe, where are you from?
I'm from Nigeria.
You're going to be in the panel next year.
Okay. Yeah. But I haven't done genetics, so probably we'll talk after. Just interested in it, because linking everything, diabetes, genetics basis of disease is quite interesting, so that's why. And I'm interested in the connection, the neurodegeneration bits, because we just begin to see all sorts.
Yeah. Well, for the juvenile glaucoma, pediatric glaucoma, this actually had been a major concern. In Boston, there was a group at Massachusetts Eye and Ear that was studying this decades ago. And frankly, the gonio cutting of gonio, that was the first group of patients that it was used on to go into the trabecular meshwork and cut the trabecular meshwork. And as you know, that for adults, that led recently to the trabectome, where it's cauterized. Mm-hmm. Other methods now going back to that, where there's no cautery, and now MIGS is coming up, MIGS, to bypass, and all of that assumes that glaucoma, in this case your question is juvenile or congenital, that it is really the trabeculum that is the problem and not the pathways beyond that, the collection channels. And unfortunately, that's when we have to have the invasive surgery because of the collection channels to drain it to the outside. And in the lecture that preceded this course, they were talking about all the complications with the drainage procedures. Too much healing at first, not enough with leakage, infection. So the nightmares when you're trying to attack and work with the poor collection channels.
Thank you.
Any other questions? Well, thank you all for coming. And I congratulate you for sticking here until the end. Yes, sir.
Just one more comment. We did a study on glaucoma on 100 patients, and we found a strong correlation between MTHFR gene and glaucoma. So this is a gene which is quite prevalent in this area. So we could find a good correlation between glaucoma and this. And so one last question. We have been talking about cornea, glaucoma, retina, and everything. And as we all know, cataract is an age-related disorder. But we do see a lot of people who are developing cataract at an early age, let's say 40, 50, and those who do not develop cataract even at the age of 80, 90. So has enough work been done on senile cataract? Why people are developing cataract at a very early age? I don't think the environmental factors are really responsible for that. I think there is a strong genetic basis for developing early cataract and maybe late cataract.
I find that some aggressive surgeons say, I will prevent you from having glaucoma. I'll take out your cataract. Give it more room. And I've seen that in my own area, and it's sickening. Mm-hmm. Let us treat the disease process and not something else. True, a swollen lens, a real cataract, a fat one, can shut off the outflow where you can get angle closure. But otherwise, keep your hands off it.
No, I was just asking about, is there any genetic basis for senile cataract? Because we know that there are multiple genes which can be responsible for cataract. Has enough studies been done on determining that why some people develop cataract early and why some develop cataract late? without any emphasis on glaucoma, just on cataract.
That's a very good study to see what the genetics of that might be, the true cataract causation of glaucoma,
Yeah.
the leakage, the fattening of the lens, blocking it. Yeah, if there's a true reason for the cataract being implicated one way or the other, go for the cataract.
Thank you.
Genetic-wise, we'll see if that could be altered. The future is coming fast. We're at the cusp of a new type of medical treatment. It's going to be genetic. Thank you all for coming. See you next year. Please, if you want a copy of this, the genetic, I will send it out in 2 weeks. Here's a pen. Give me your email.